Our first speaker for this session is Dr. Tal Gilboa from Weiss Institute, Howard University. Tal.
Okay. Do you see my screen?
Yes.
Now? Yeah? Okay.
Yes, all good.
So first, I want to really thank the organizing committee. Thank you so much for your hard work, and unbelievable how it actually happened at the end after a year and a half. So today I'll just show you a little bit about our work towards the quantitative diagnostic and staging of Parkinson's disease. One sec. Okay, and so currently when a patient starts having symptoms for Parkinson's disease, they will go through a series of tests, see a neurologist. Sometimes they require some imaging until the clinicians reach a subjective clinical diagnosis, because currently there are limited tools to diagnose Parkinson's disease. And this can take a while until they start treatment to minimize symptoms and slow down progression, because there is no cure for Parkinson's disease. And the reason we cannot diagnose these patients earlier, one of the reasons, is that we cannot see what's happening in the brain of living patients. We can just biopsy the brain easily. But if we could, we would see that neurons are dying. And so the hallmark of Parkinson's disease is the misfolding and aggregation of this protein called alpha-synuclein, resulting in Lewy body and death of the dopaminergic neurons. But actually, symptom onset is already too late. It's already when the majority of the specific neurons died. And the initial event which starts this disease can happen even decades before symptom onset. And so we do have a very precious window for intervention if only we could diagnose these patients earlier, identify new drug targets, help clinical trials, and hopefully develop cures. And we can think broader than Parkinson's disease about how we currently learn about the brain. So we mostly know things from animal models and postmortem brain tissues. And so there is a gap in the methods available, and there is a need for technologies where we can really see what's happening in the molecular level in the different cell types in the brain of living patients. But we do know that all the cells, including neurons, secrete molecules to the circulation. They're secreting DNA, proteins, small molecules, and also these nanoparticles, which are called extracellular vesicles. I'll refer to them as EVs in this talk. And these EVs are released from the cells. And so on their surface, they have specific markers from the cell of origin. And as they come out, they capture inside, they basically sample the cells and they capture inside proteins and other biomarkers and preserve them in the way they were in the brain. And so our goal in this project was to basically fish out these EVs coming from the brain and then burst them open and look at different alpha-synuclein isotypes and biomarkers to really diagnose and monitor disease progression in Parkinson's disease. That was the main goal, and it had to include lots of different methods development. So first, how do we isolate EVs from clinical samples, starting from, let's say, 1 ml of plasma in an automated high throughput way and a reproducible way? How can we really find these neuron-specific markers that we can use to fish out these brain-specific EVs? And then when we have the EVs, how can we really monitor and detect these specific alpha-synuclein isoforms? And they're going to be very low level because the majority of the proteins in the EVs are not from the brain in the plasma. And so I'll just show a few of the things we've done to achieve these goals. So first, we thought about how do we optimize our EV isolation, specifically for clinical application. And when we compare different methods, we found that using size separation is actually very efficient. So here we have these columns where we optimize a specific resin inside, and this resin has small pores such that the EVs will just go down and elute first, and the small molecules like proteins will be trapped in the pores and will elute later. And we have automated the EV isolation using these SEC columns. And here you can see our octosome where we have a Raspberry Pi computer connected to a pump, and this pump can flow our PBS or other buffer and move between a waste and sample collection. And we've optimized the columns. And so we have columns which are better for high yield EV isolation, getting the majority of the EVs out of the clinical samples, and columns to allow us to isolate more pure EVs. For example, for biomarker discovery. So here we combined size exclusion separation and some cation exchange because the EVs are mostly negatively charged to get very clean EVs, and we established this proteomics of EVs in clinical samples. But that was just 8 samples, and for clinical studies, we want to look at maybe hundreds of samples. And so we've developed these high throughput EV isolation methods where we have these plates look just like a regular 24 dipole plate, but actually it's different because each well is an SEC column, which performs just the same as a regular column. And now when we have this plate format, we can integrate these plates with automation robots, designing adapters and the hardware to do that. And so here we showed how we can automatically isolate EVs from 50 samples completely automated. And so now when we have these platforms, this is just for isolating all the EVs from clinical samples, but we want to see specifically if we can isolate these neuron-specific EVs, so moving from total plasma EVs to neuron-derived EVs. And so we used our EV proteomics data and combined that with data sets looking at what proteins are transmembrane, because we want to use this transmembrane protein to fish out our brain-specific EVs. And we looked at gene expression data and looking for markers which are brain-specific within the body and neuron-specific within the brain. And we identified around 28 markers, which we know are going to be very neuron-specific EV markers and also are transmembrane, so we can use them to fish out these EVs. And we complemented this experimentally with a few different methods, which I'll not describe today. First, we can look at size separation of our target molecule and see if it elutes with our EVs or with a later fraction with the free proteins, because some of these markers can be cleaved in the blood. We can do colocalization using single EV imaging to colocalize our neuron markers with other EV markers. And we've optimized pull downs to really efficiently pull out these EVs using our targets. And we have all these methods. Our goal is to basically open these EVs and measure alpha-synuclein. And so to do that, we've developed single molecular array assays. It's a method developed in the Walt lab. It's basically digital ELISA, very sensitive ELISA assays, where we showed how alpha-synuclein, when we just fractionate plasma samples, has a 2-peak distribution. You can see in the bottom in blue. So the first peak elutes early, and you can see how it overlaps with CD9. It's a transmembrane protein. So it's what we call an EV-associated peak, whereas the second peak comes later. So it's just smaller alpha-synuclein protein circulating in the blood. And we can use this data to calculate the ratio of alpha-synuclein in EVs. And we saw how around 5% of the alpha-synuclein in the plasma is in the EVs. But the question is it actually inside or just stuck on the outside? So we can digest the external protein using ProK, then inhibit it and burst the EVs to measure the internal alpha-synuclein in the EVs. And the majority of alpha-synuclein in the EV fraction were actually inside the EVs. And so now we know that we have alpha-synuclein in the brain. This was an assay for all the alpha-synuclein, and we're specifically interested in what we call a pathological alpha-synuclein seed, this misfolded alpha-synuclein that will cause this aggregation in the brain. So how can we detect this specific pathological alpha-synuclein form? So for that, we tried to mimic what's happening in the brain of Parkinson's patient, just in a very, very small compartment. And so we developed these digital assays where we take a brain sample, or in the future, blood sample after our EV isolation. And if this sample has the seed, which is the pathological alpha-synuclein protein, and we add more alpha-synuclein to the sample, it will basically recruit the alpha-synuclein and starts to aggregate. And so we can take this solution, add this dye, which is specific for amyloid fibers, and confine each single seed into a small compartment. It can be a droplet or a microwell. And then you can see if we have seeds in the sample, they will start forming aggregates over time in the small compartments. And so we can really monitor the aggregation growth, and we can count the number of on wells or wells with signal. So if we take a clinical sample and we dilute it, you can see how we have less and less wells with a signal, showing how this assay is very quantitative. And so now we have lots of methods to detect alpha-synuclein, and we have the isolation, and we have our neuron candidates, and we're in the process of combining all these puzzle pieces together to basically allow this non-invasive blood-based test to look at alpha-synuclein over time in Parkinson's patients. And our goal is to develop new tools for biomarker discovery, to find new targets, to enable clinical trials, basically to replace very expensive imaging, and have better patient selection for clinical trials. And in the future, hopefully, when we have cures, we'll also have a detection method. When we're 40, 50, 60, 70, we can just have a screening test for Parkinson's disease. And lots of people to thank. So we have the Walt Lab here and the Michael J. Fox Foundation, which are supporting all of our research, our other funding, the Science Abroad, and specifically Nest Catalyst program, which helped me to start our company, Everest Biolabs. I'm going to be joining Tel Aviv next summer. So I would love to speak with any of you about Parkinson's, single molecule methods, exosomes, or any other related topics. And thank you all.
Great talk, Tal. We now have time for some questions. Anyone from the audience? Okay, I'll start then. Were you also able to detect the phosphorylation of alpha-synuclein in the exosomes?
So I didn't have time for everything, but one of the things we've developed are assays for specific phosphorylation as well. And we actually showed how in the EVs we have enrichment of phosphorylation. So what we think is happening is that the EVs are protecting the proteins. So for example, protecting alpha-synuclein from phosphatases and other enzymes in the plasma, and we're also doing that for other proteins as well. So I think, yes, great question, and phosphorylation is very interesting in terms of EVs and how they protect it.
And I also have a question.
Yes.
Great work and very, very interesting for me. And I was wondering about other synucleinopathies and other cells. So your approach is focused on identifying neuronal EVs in Parkinson, and very impressive work. What about other synucleinopathies, such as MSA?
MSA, yeah.
And
awesome question. And
yeah,
great question. I did not have time to talk about it. We have in the paper actually very nice data, how we are showing that the MSA and the Parkinson seeds look differently in our microvalves. And we are looking for targets for oligodendrocytes, astrocytes, and other cell types. So the dream is to fish out all these different cell types and look at what's happening in all of them over time. That's the future goals. But yeah, definitely it's something I'm very interested in.
Okay, any other questions? Thank you, Tal.